Precision Nanosystems Transfection Kits encapsulate nucleic acids in proprietary lipid nanoparticles that mediate high transfection efficiency in difficult-to-transfect cells, in vitro and in vivo. Our lipid nanoparticles efficiently transfect primary cell lines and stem cells by exploiting the low-density lipoprotein receptor endocytosis pathway. Nanoparticles are applied to cells in culture. Apolipoprotein E, or ApoE, adsorbs onto the surface of the lipid nanoparticle. ApoE is a ligand for the LDL receptor. As LDL receptors are ubiquitously expressed on primary cells, this mechanism can be exploited to promote nanoparticle uptake in a wide variety of cells. Endogenously circulating ApoE facilitates uptake of the nanoparticles in vivo, while nanoparticles transfecting cells in culture may require the addition of exogenous ApoE. The lipid nanoparticle ApoE complex is recognized by the LDL receptor. Endocytosis takes place, and the receptor particle complex is invaginated by the cell to form an endosome. As the endosome matures, internal pH changes promote nanoparticle lipids to interact with endosomal lipids and release the nucleic acids into the cytoplasm. Neuro9 Transfection Kits are engineered to prepare lipid nanoparticles that transfect greater than 95% of cells, mediating 95% gene knockdown in vitro, sustained for more than 21 days, and greater than 80% knockdown in vivo, maintained for at least 15 days. By utilizing endogenous uptake mechanisms, Precision's engineered nanoparticles enable high transfection efficiency with low toxicity, ideal for functional genomics and drug biomarker target validation studies in primary cells or induced pluripotent stem cells.